Okay, good morning, everybody. I'm glad to see you all after the long weekend. So I thought we'd start today with a review of last week since it's been several days since we've talked about this material. So I hope that's okay with everybody. I'll try to go through sort of quickly of this so we don't get too bored of it. Okay, just to give you an overview again of the course and some of the things we've already talked about. We've talked about a little bit of an introduction to neuroscience and to the nervous system. We went over some brain anatomy. We talked about mapping function to structure and some anatomical and functional neuroimaging methods, and we went over MRI. And so now we're at functional MRI. So we talked a bit on Friday about the Bolt signal, how it works, design of experiments, and data processing. Today we're going to go over more details of how we model the data and some of the statistical problems, and we'll finish up the week with MRI safety, functional connectivity and resting state fMRI, and diffusion tensor imaging, and we hope to get into some applications of this technique as well. Again, the course expectations are just to get a basic overview of the concepts here and gain a basic understanding of the brain and the nervous system, some imaging techniques, the extensions of these techniques, and some applications. So again, please don't be afraid to stop me and ask me questions, or if I'm speaking too quickly and you want me to slow down, just please raise your hand and say something. Okay, so if you recall from last week, we talked about the early methods of brain exploration and how limited they were. There were animal models, which still happen today, but they're often difficult to apply to human techniques, human applications. We talked about postmortem dissection, and then we talked about unfortunate diseases or malformations or accidents for some, while the field of understanding the brain benefited from these. But these are things that happen by chance only, so it's not an ideal thing to wait around and for someone to have an accident so we can understand their brain. So as we went on, we saw how some of the imaging techniques of the 20th century started to improve the methods and the ways that we can study the brain. So if you'll recall, we talked about the nervous system, how we have the central nervous system, the brain and the spinal cord, peripheral nervous system, which is the part of the nervous system that's not enclosed in bone. It goes out throughout the body. The peripheral can be divided down into the somatic nervous system, which we have our sensory neurons, the afferent neurons that come in and bring information into the central nervous system, and then we have the efferent neurons, the motor neurons, which go out and allow us to move and interact with our environment in response to sensations. We talked about the autonomic nervous system, which is the part of the nervous system that controls internal organs and different glands in the body, helps us to breathe. And the autonomic is the part of the nervous system that we don't mentally control, that goes on in the absence of any thought, and it really sustains us throughout our life in doing all the vital functions that we need. And this included the sympathetic nervous system, which is the arousing part. So if we get in a sticky situation or need to react quickly to our environment, this is the part that activates, whereas the parasympathetic is more of a calming part of the nervous system that activates when we are relaxed, had a big meal. It helps in digestion and functions that aren't related to acting quickly within our environment. We talked about the cells of the nervous system, the neurons, which are the cells that send and receive information. And these have the dendrites, which receive signals, and the axons, which help to pass along the signal throughout the cell and to other cells. We talked about glial cells. These are the most abundant cells in the nervous system, and these are thought to be cells that support structure of this nervous system. They're not the cells that send and receive signals, so they don't conduct the electrical signals, but they perform other functions within the nervous system, such as myelination of the cell axons, making the cerebral spinal fluid, creating structure for axons to grow on. So we know these are important cells as well. We don't understand their function as well as we do the neurons. We talked about neuron communication. So these can have communication within a cell. This is when a cell receives information and propagates the information along its axon. And we talked about intercell communication or neurotransmission, and this is when a cell sends an information to another cell across the synapse that connects one cell to the next. And if you recall, these synapses are connected to each other through a chemical process rather than an electrical process. We talked about myelination, which helps for quicker cell conduction within a cell, sort of the nerve conduction. This is fat cells that wrap around the axons and leave little nodes open so that the signal can jump from one node to the next rather than taking the time to have to do the action potential all the way along the cell. So this increases the speed that we can send information through a neuron. And again, this is the tissue that looks white and is within the brain rather than the gray matter that is mostly on the surface of the brain. So this white matter allows cells to communicate quickly, and this is a lot of what we see when we look at the brain in images. You can see on the left is the gray, on the right is the white matter. So we talked about the planes of the brain. Axial plane, cutting this way. Coronal plane, which is cutting off the face, I think of. And the sagittal plane, which cuts down symmetrical to the two halves of the body. The sagittal plane can also move this way and that way, but I try to remember it by sagittal symmetry. So it's the plane that if you cut in half, you get two symmetrical sides if you cut it right down the middle. And if you look at these images here, these are T2 weighted images. We can tell because the cerebral spinal fluid is white in these images. And we'll briefly go over again how we use different tissue properties to make different contrasts during an MRI. I know we went through that pretty quickly last week. So we talked about the brain and the parts of the brain. So the brain, of course, is a large mass of nervous tissue. It has four main parts: the cerebrum, the limbic system deep within the brain, the brainstem, which contains a lot of our autonomic functioning, and that's where a lot of our breathing and things are regulated, and the cerebellum, which is the little brain in the back there. Cerebellum means little brain, but it's that part that sticks out the back. We talked about the lobes of the brain and the parts of the cerebrum. We talked about the sulci and the gyri in the brain. Those are those grooves and those valleys that make up the gray matter on the surface of the brain. We talked about fissures, which are the larger grooves that separate the different lobes of the brain. And we talked about the frontal lobe, the occipital lobe in the back, the parietal lobe, which sits up here, and the temporal lobe, which has a left and a right side. And we talked a little bit about the functions of each of these lobes. Then we talked about the limbic system in the midbrain and how this is important for a lot of our primitive functions, or what we would consider today to be primitive functions. This is located deep in the center of the brain. Main parts being thalamus, hypothalamus, amygdala, and hippocampus. And as part of this limbic system is known to support the emotional processing in the brain. It also contains other functions like behavior, helps with long-term memory in the hippocampus, and olfaction, which is the smelling center of the brain, which comes from the frontal lobe at the bottom and goes into the limbic system. Then we went on to talk about the different functional systems in the brain. So these are the systems that work, or the anatomical regions that work together to help us perform different functions. And you see here some of the things that we talked about, and how that none of these systems works exclusively or independently. They all work together, and they integrate the information to help us make sense of the world around us. We talked about sensory processing, which in all cases doesn't matter if it's tasting, smelling, hearing, or seeing. It starts outside the nervous system at the location of the sensory organ, and is amazingly transferred into neural signals. So when we see something, the vision goes to the back of the retina. The retina converts this light into neural impulses, and these are sent initially to the primary processing center. For example, the primary visual center in the brain, which is in the occipital lobe. After the information reaches this place, it's then passed to the secondary processing center in the brain, and from there it goes oftentimes to the parietal lobe, where it's all put together and helps us to make a complete picture of the world around us in the brain. We talked about top-down versus bottom-up processing. Bottom-up referring to the processing of sensory information and some of the more primitive functions that we have in the brain. These include emotion and fear processing. These types of inputs that come from bottom up are usually externally driven. So we see something, we react to it, or the information comes from the bottom of the brain up, versus top-down processing, which refers to processes that are driven more by perception and cognition. So these include executive functionings and controls, and this comes usually from the prefrontal cortex, and comes down sort of to control our reactions and how we respond to the sensory information that's coming up. So this top-down processing, part of this is we refer to as executive cognitive functioning. And you can see the picture of the brain here, and how the front of the brain comes and performs a lot of functions from the front back, including helping us to keep our attention on things, helping us to inhibit responses, helping us to regulate our emotions, helping us with working memory, problem solving, and planning and effort. So this is the part of the brain that helps to distinguish us from less developed animals, or what we think of as less developed animals. And this is a part of the brain that really makes us human beings and helps us to control behavior and regulate ourselves. So again, these are essential for daily living and mostly occur in the prefrontal cortex. So we briefly talked about the reward system, which comes from the midbrain, the ventral tegmental area, and nucleus accumbens, which are the groups of neurons that sit low inside the midbrain. The reward system helps us have incentives and desires. It helps to reinforce positive behaviors, and it includes a lot of the dopamine-rich areas that are in the brain. It helps us, it's sort of located between executive centers and the sensory centers. We talked about emotion and emotional regulation. The affective center of the brain, which is part of the limbic system, amygdala, thalamus, hippocampus, and how the orbital frontal cortex, which is more of the bottom of the brain here, it's also part of this center is also referred to as the ventral medial prefrontal cortex, which is why it says VMPFC. This helps us to regulate our emotions, and within this emotional center also is located the fear centers, which helps us respond to emergency situations in our environment. And then the frontal cortex comes along and sort of helps us to know if these fear responses are warranted or not. So we can become accustomed to something in our environment that scares us, but we know isn't a huge threat to us. We talked a little bit about short-term versus long-term memory. So we talked about how sensory information is constantly going on around us, but it's quickly lost if we don't attend to it. And this is probably a good thing, because if we try to absorb and remember everything that went on around us all day long, we would be on overload with our brains. So we talked about short-term memory, how we can hold some items in our brain for about 20 to 30 seconds before we lose it. And the way that we can retain this information is to transfer the information to long-term memory. So the things that we feel are important to us, we tend to keep in there and send it to the long-term memory systems, which can hold information indefinitely and hold unlimited capacity of information. Or that's what we think anyways, now in terms of our research. So we talked about how the 1900s brought these new techniques in the 20th century. EEG, we talked a little bit about x-rays and CAT scans, and we talked about MEG, or magnetoencephalography. Then we went on to talk about MRI, which we'll review briefly here, and PET scanning. Then we went on to functional MRI. We haven't yet talked about diffuser tensor imaging, but we will talk about that hopefully tomorrow. We talked about structural versus functional imaging. So structural imaging being designed to see anatomical features of the tissue. So these include x-rays, CAT scans, MRIs, and the diffusion tensor imaging. And these are highly and often used for clinical assessments to try to look if something is wrong inside the person's brain anatomically, if they had some kind of a damage or bleed, if they have a tumor or abnormal growth within the brain. And in research, we often look at structural integrity of the tissue and try to compare the sizes of different parts of the brain in different types of populations to see if something in the volume is different, for example, in people who are violent versus people who are nonviolent. Then we talked about functional imaging and how rather than seeing anatomy with this, we look for function with this. So we talked about functional MRI and PET scans, EEG, MEG, and NIRS we didn't talk about, but these are all functional techniques. So we're not looking specifically to look at the shape or size of things in the brain. We're trying to understand how the brain is working when we look at these types of techniques. These are used for clinical assessments to a lesser extent than structural, but they're widely used in research. And as the techniques develop, we may tend to use them more and more in clinical assessments as people gain a better understanding of them. At this point, a lot of the techniques are very new, and we're not quite sure how to apply things like functional MRI to a clinical assessment. Then we talked about, with our functional techniques, the difference between direct measures and indirect measures. So direct measures are actual measures of the neural activity, or what we think is an actual measure of the neural activity. This includes EEGs and MEGs, and these are signals that are recorded from the scalp, typically, or in some cases from the brain directly. These give us great time resolution, but not so great spatial resolution, especially if you're looking on the scalp rather than directly on top of the brain. And we talked about indirect measures. So these are measures of the side effects of this neural activity. So, for example, PET is a measure of metabolic processes, or processes that are going on within the brain, such as glucose metabolism, blood flow, different levels of neurotransmitters that are released in response to different processes or different functional states, such as somebody taking drugs versus somebody not taking drugs. And we talked about functional MRI, how this is actually a measure of changes in blood flow activity and oxygenation levels that are distributed locally within the brain. So we went a great deal over EEG and the principles of EEG, how these are, again, thought to be direct measures of neuron firing, and these are actually summations of the processes going on underneath the scalp. So we are not making assumptions that we're looking at single neurons or groups of neurons. We're just looking at the specific location of the head, the underlying electrical activity that's happening there. The activation from EEG is mostly from the cortex, because this is what's closest to the scalp. Although the slow wave activities in sleep are thought to be originated lower in the brain and in the deeper centers of the brain. And again, how it has excellent time resolution, but not so great spatial resolution. We went over this 10-20 system of electrode placement. And on Friday, we had the wonderful opportunity of going to the lab and seeing how an EEG cap was placed and how this 10-20 system works. You can have either a cap that helps with the placement, or you can have a tape measure and have someone measure all these spaces of the electrodes. But they're placed in a standardized way, and the different locations are called standardized things internationally, so that everyone doing EEG research understands where an electrode and the signal that was measured is coming from. And so this is a great way of trying to make EEG research applicable and standardized throughout the whole world. We talked about how EEG has evoked potential studies. This is when you elicit a stimulus to somebody and then have see what the signal is underneath in response to this. And the thing that we looked at was looking at the alternating checkerboard and how this elicits a very expected visual evoked potential in the occipital lobe. So we talked about how people look at that and the latencies or the time components of that signal to try to understand how quickly the information is being spread through the neurons. We talked about this time frequency analysis where we looked at the Fourier transform, and you can see in the picture here how frequency waves are distributed over the brain, and we saw a little bit of that in the lab when we went Friday as well. We talked about this self-regulation thing where people look at their EEGs and try to change it just by doing different things mentally. And we talked about source localization, about how people are trying to understand the origin or the main generator in some of these signals underneath the surface of the brain. Then we talked about PET imaging. This, again, is a non-invasive diagnostic imaging technique, and it measures metabolic activity of the cells depending on what type of process you're looking at and depending on what kind of tracer you use. The tracer is called a radionucleotide, and this is introduced into the body on a biologically active molecule. So remember, we said PET was non-invasive, but non-invasive because we are using ionizing radiation. So we're injecting these positrons, which are positively charged electrons. Again, whenever we have a biological process, these are released. They collide with a normal electron and annihilate. So it's antimatter. When it collides with matter, it annihilates, and it sends a pair of gamma rays exactly 180 degrees apart at the same exact time, and we use a little ring around the person. And when this ring detects simultaneously occurring waves hitting these detectors at exactly the same time, then it records this as a signal. Again, here's the idea of these PET tracers. So you label some kind of compound with a radionucleotide. These go into the body, they go through their biological event. For example, FDG is a tracer that mimics the effects of glucose. So when FDG is metabolized, this tracer is released and goes off and does its annihilation with the electron and produces a signal. Okay, and then we went on to talk about MRIs. This is the high resolution anatomical imaging. And we talked about all these different things: magnetism, nuclear spin, precession, the RF pulse, the free induction decay signal, T1, T2, T2 star, and how we get contrasts in the image using these T1 and T2 properties of tissues. So I'm just going to go briefly through this again. I hope you guys aren't too bored. So I always think that repetition is one of the best ways to learn something. So I'm just going to go through this quickly again. As you put hydrogen atoms into a magnetic field, they have an odd atomic charge, or nucleus. They have an odd atomic number. So they have a magnetic spin, and when you put them in an external magnetic field, they all line up within this field. Then we hit them with this RF or radio frequency pulse, and what this does is it tips them down into the XY plane. So if they're lined up within the Z plane, we tip them down with this RF pulse and knock them into the transverse plane. Then we let them do their thing to get back into their lowest energy state, which will be lining up within that magnetic field again. So two things happen after we hit them with this pulse. One is that they immediately want to go back to being lined up in this magnetic field. This is called T1, longitudinal relaxation. And as they do this, they emit energy. And the second thing that happens is this loss of phase coherence in the XY or transverse plane. So when we initially knock them down, they're all together in a phase-wise, and then they start to separate as they go back into the field. So again, the signal that we measure while they're doing this is called the free induction decay signal. And this is what we're measuring as they're returning to their initial energy state or their lowest energy state. So this is an oscillating signal that we measure with a receiver within the radio frequency coil. So again, T1, we're talking about this longitudinal relaxation or spin lattice relaxation. T2, we're talking about the loss of phase coherence or spins between these as they process and go back into their initial state. T2 star is the loss of phase coherence due to the inhomogeneous magnetic fields. And T2 is actually a product of T2, which is the perfect T2. So the T2, that's actually a tissue-dependent property, what we would see if we had a perfect magnetic field. And T2 star is what the T2 is caused by the imperfect field. So T2 is actually an addition of both T2 and T2 star. T2 star causes faster decay in the signal, so it causes the T2 to be faster than what it normally would be in a perfect field. T2 star is what we use to measure functional MRI, and we'll see this again very briefly. Again, there's T2 and T1 are different for different types of tissue. So in solids or in bone, the T2 is faster because the molecules are closer together. So they're interacting together to cause this faster dephasing or the faster separation of those phases as they spin together in the transverse plane. T1 is how, again, influenced by what kind of tissue there is. So you can see that more viscous materials have a shorter T1, and materials that are not as viscous, like CSF, cerebral spinal fluid, or water, have a longer T1. Okay, so I thought we could benefit from talking about T1 and T2 tissue contrasting again. And so we're just going to go over this briefly. And I found these nice pictures on this site called MRI Shark. So here they are. And what I wanted to point out here is how we get the contrast in when we do T1 tissue contrasting. So when we first flip a slice of tissue down into the XY plane, these what's actually going down is the hydrogen atoms, because we tip at the resonant frequency of these. So if you remember looking at the child on the swing, the resonant frequency is the natural frequency that the child is going back and forth on. So if we push these at that, or if we push the child at the frequency that he's going, that adds more energy into him. So, but if we try to push him at a different frequency, what's going to happen is the swing's going to go different ways and go haywire. So we actually tip them at the resonant frequency of hydrogen, and then as he's relaxed back into the field to try to line up in the field, we can use the fact that different tissues relax at different rates to make this contrasting. So if you look at the plot here, hopefully you can see it okay. Initially, when we tip them, all the tissues are at the same state. So then as they relax back, different tissues will have different speeds of relaxation. So what this is showing here is what the images would look like if we did T1 contrasting at different points along this tissue recovery. So the first line shows what happens if we, in the corresponding image and underneath, if we would collect the signal, or basically that free induction decay signal, we listen to it with our receiver too early. So you see there's not a lot of contrast between those tissue types. Now, if we wait till these start to recover, then we start to see a separation in the tissues because the different tissues are going at different rates. So the second image here shows if we wait about 200 milliseconds, we see some T1 contrasting, but it's not ideal. So the third tissue would be our optimal T1 contrasting. So this is about when 63% of the magnetization has been regained. So this is about at a point where about 63% of all our ions have moved back into are starting to move back into that alignment again. And we can see here that this is the optimal time for us to measure. The cerebral spinal fluid comes out black in this T1 contrasting, and I actually told you that white ring was bone. That is incorrect. That is fat around the head. The myelination of because fat has a very fast recovery. Fat is actually that yellow line, so it has the fastest recovery compared to the other tissues. The blue line, I mean, that goes on the bottom there is cerebral spinal fluid, and water also has the longest recovery of all the tissues. And again, so this turns out black. The tissues that have a faster recovery, such as fat, turns out white in these images. And if we wait a very long time, the T1 contrasting is fading in this last image. So T1 contrasting is with a short TR, so that's a time of repetition, how quickly we're flipping the atoms as we take the images over and over, and a short TE. So we want to flip quickly. We don't want to wait too long, because if we wait too long, everything's going to be recovered back into that plane. T2 tissue contrasting, on the other hand, is due to this loss of phase coherence. So T2 contrasting has a longer TR and a longer TE. And the reason for this is because when we first flip these protons or hydrogen atoms down, they're all together. So they're all phase locked together. But as we wait, this phase locking or phase coherence starts to decay, and they become less coherent with each other. So if we wait a longer time, then we see more of this loss of phase coherence. And again, this shows what happens with T2 contrasting as you go along. So on the far left, the T2 a lot of the phase coherence hasn't yet been lost, so there's not a good contrast in the tissue. Whereas if you go along to about the third image there, about 63% of the transverse magnetization has been lost, and this gives us our optimal T2 weighting. So in this case, water or CSF has the slowest phase coherence loss. That's that blue line again, whereas the fat has the fastest. And so in this case, because the CSF is the longer tissue for phase coherence, in this case, the fluid comes out white. So if you see white CSF in an MRI image, you know it's a T2 weighted image. If you guys want to look up more details about these, this website I found called MRI Shark had some great explanations as to the differences in these types of contrasting between tissues. Okay, then we talked a little bit about slice selection, how we can add a magnetic gradient, and this will change our laminar or a resonant frequency. So to get spatial resolution, we can change the magnetic field slightly in the slices, and this will allow us to distinguish slices of tissue from each other. Similarly, we can apply different gradients in the X and Y directions to get spatial information in our X and Y plane. Okay, we talked about pulse sequences, and we talked about spin echo. So we're not going to go over that again today, but that's that technique where you flip and then you flip 180 degrees to get the phase coherence to converge again rather than diverge, and you can get rid of the T2 star effects using this and get an optimal image. And we talked about the faster techniques like echoplanar imaging, and this is what we use for functional MRI. So we flip all the protons and we measure the whole slice at once by adjusting these X and Y gradients in a similar way that we would adjust the knobs on a labyrinth, so that we can sort of direct where our signal is going and we know what we're recording spatially. Okay, we talked about K-space and how this is the raw image space in MRI. So what this is again, we're measuring these oscillating frequencies that are coming, and it's a measure of how much of each of these frequencies is contained in our image. So if we do from K-space a Fourier transform to get into image space, a similar way that we would do it in one dimension, where we're trying to fit sums of sines and cosines to a time signal. Now we're doing this in 2 dimensions, so we're trying to fit, again, sine and cosine waves to make a 2-dimensional images. And instead of these looking like a time thing that we would see in 2 dimensions, now we can look down from 3 dimensions, and what it looks like basically is stripes of black and white. So we can use the stripe information by putting different weights to the different frequencies and the different intensities. And this is what makes the picture of the image here. We talked about low frequency in K-space versus high frequency. So the middle picture here is the ideal image that we would get if we took into account all the low and high frequency information. If we chopped off the higher frequencies, which are located on the outer bands of K-space, we get a low frequency image, which allows us to get the bulk of the detail of what the image is. But the high frequency information, which you can see on the right, can you guys see those black squares in there? Okay. We're blocking out the lower frequency information and only looking at the high frequency. And what you would get in this case is the detail of the image. So ideally, we want to have our MRI signal cover enough of this space so that we can get both the bulk information of the image and the detail of the image. Okay, then we went on to talk for a long time about functional MRI and how functional MRI uses the blood oxygen level dependent or bold response, or the bold signal is what we call it. Functional MRI, compared to the techniques that we've used in the past, has a really good spatial resolution and a fairly good temporal resolution if we can collect our images fast enough and model this data accordingly. So functional MRI is completely non-invasive, so we don't have to use any ionizing radiation. We don't have to use any dyes or tracers. So it makes it really ideal to study different clinical populations and different types of research problems that we want to address. The studies are fairly easy to implement. So if you remember, the basic concept of the bold is that when a tissue is at rest or at baseline, we have the blood coming through and some of the blood is oxygenated and some is not oxygenated. This means that the hemoglobin molecules within the blood on the red blood cells are carrying iron to the tissue, and it's these local changes in the levels of oxygenated versus deoxygenated blood. And so these basically these iron amounts change in the tissue when the neurons activate. So when they activate, their metabolic demands increase. The body responds by sending more oxygenated blood to these areas. And this iron on the blood changes, and this changes the magnetic field slightly because iron is a ferrous material that is attracted to the magnetic field. So we see local changes in this field, and this changes our T2 star effect just slightly. So we see these very small changes in local areas that are using more oxygen. So this is the whole concept behind functional MRI, that when our neurons fire, they need more oxygen, and then this changes how much iron we can see in the little local areas, and how the magnetic signal is affected in those areas is what we want to measure. We talked about how the bold signal is also the hemodynamic response function. So it's a measure of the blood flow sort of in response, or in what is contained in that blood in response to our tissue needs. So this is a slower signal. The body, if we have a stimulus here in the red, the body then changes the blood again, the blood oxygenation levels in the area, and we can measure this bold response, which takes place about a period of 15 to 20 seconds, with a peak occurring about 6 seconds after we use a certain area of the brain. Okay, we talked about how we measure this a little bit. So we have a person alternating between two states while they're in the MRI machine. So for example, we could have them just rest, or we could have them tap their finger. And while we're doing this, we're recording these slices of tissue very rapidly over and over again. And then we take the slices of tissue that are related to when the person is at rest, and we take the slices of tissue that are related to when they're tapping their fingers, and we do some statistical analysis to see where in the image the intensity changed and then we can use this to determine where activation occurred in the brain. So remember, functional MRI data is very low resolution compared to a structural MRI data. And remember, we're not measuring the amounts of hydrogen in the tissue with this technique. We're measuring the bold response. So we need to collect the data very rapidly, and because of this, we're losing our nice spatial or structure that we see in the tissue, and we're sort of getting these chunky blocks of intensity, and these little blocks are called voxels. So they're a three-dimensional pixel, so they're a volume element or a voxel. Okay, we talked a little bit about designing experiments. So we put a person in there. We want them to do something simple that they can do while they're in an MRI machine. The environment is limited. It's a very confining little space. And so we don't want them having to do anything complicated that they're going to have to, that would make them move around. We want them to stay as still as possible. The machine is also very loud, so we have them put in earplugs. So we're limited to what we can do in terms of auditory signaling. We want to, again, consider the timings of our experiment, because we want to have enough power when we do the statistical analysis to be able to see these small areas of activation. And usually we want to keep track of how well the person is performing while they're doing the experiment. And this is a measure of what's called the behavioral data. So we can have them pressing a button, as we saw when we looked at the implicit emotional processing. We said, press your finger if you see your index finger for female, middle finger for male. So we use this behavioral data to make sure that they're attending to the task. And we can also use this data to see how well that they're doing in the task. And we could even use this measure of performance maybe as one of the factors that we put into our model when we analyze the data. And we're going to be looking more specifically at data modeling today. So remember, when we design an fMRI study, we want to do something similar and only change one variable. So we might have them look at a crosshair, or we might have them look at those bathroom figures versus, the man and woman figure versus looking at emotional phases. So we want these regions that are very prominent in activation, for example, using the visual system on the right here, to cancel out as much as possible, so that we don't have the tasks so differently that we see, for example, doing auditory tasks versus visual, because if we try to compare these, we're going to have areas of activation that are attributed to all sorts of different things, and we can't really pick out the effect that we want to see or attribute what's actually happening to a specific area of the brain if we have the brain activating all over the place. Okay, we talked about the block design versus the event-related design. So the block design, again, is where you alternate two conditions in a longer time period, with the assumption that the more a person does a task repeatedly, the more this bold signal is going to rise and stay up until they stop doing the task. So again, the benefits of doing a block design are that you have a lot of power in your signal, because you're having the bold signal rise and stay up and then come down again. But you lose any temporal or time-dependent effects that we see in this type of data. So people moved on to what's called these event-related design studies, which they elicit the stimulus where the arrows are, and then they try to measure the bold response in a region. And if you do this over and over again, then you can stack this up, and sort of like the ensemble averaging that we saw in EEG, that the more you do something over and over again and put these together, you can make a better average of this actual signal, and get rid of the noise by assuming it's a white noise, so it's statistically random, and you hope that your noise is going to cancel out because it's random noise. Okay, so then we talked about fMRI data analysis. First, we want to process the data to remove the noise. Then we want to normalize the brains into a common space. And then we're going to do some statistical analysis of these subjects. So what we're going to talk about next today is really statistical analysis and how we generate these activation maps and what activation really is and how we see it and how we model it. So just to get us back to that point, for pre-processing or the processing steps that take place before that are a few different things that we do. For example, we are going to convert our raw data, which is the DICOM files that we talked about, to image files. So we want to have something that our software programs can read and work with. We do slice timing correction in the case of event-related design studies. This is where we take all the slices in the brain and move them back to the same starting point. So again, when we collect through the brain over a period of 2 seconds, each slice is collected at a slightly different time point. So we can get rid of this effect by moving everything back and assuming that all slices were collected at a single starting point. It's very important to look at the motion in your data and to do motion correction. If the person moves too much, then the activation that we are going to see is going to be due to this motion and not due to the blood flow changes. So that's no good. We want to minimize this as much as possible. And in a lot of cases, we throw out the data if the motion is too much. So motion is a big problem, especially in children. We did a study of children, and we ended up throwing out about 40% of our data because these younger kids have a really hard time staying still for an hour. So this is a big limitation of some of the different subject populations that we look at. Okay, then we want to line up the person's anatomical and functional brains. This is called co-registration. So this allows us to see areas of activation anatomically. So anatomically by meaning what anatomical regions are associated with our functional activation. In order to compare groups of subjects, we want to normalize all these brains to a common brain space. This is to get them all lined up so that they all can be the same size and the same shape, so we can compare, so that we can put these groups together and compare different groups of subjects. And then we can do this spatial smoothing, which again smears the information across voxels. And there are some benefits and limitations to spatial smoothing. So if you remember, we talked about normalizing to a common brain space for group analysis. So this allows us to compare brains that are different shapes and different sizes. These also allows us to compare brains that have different features such as cerebral spinal fluid volume and things like that. But we're warping the brain, so we're distorting everybody's brain. So it's really not an ideal thing to do, but for looking at groups of subjects, this is often a necessary step so that we can compare across subjects and groups of subjects. So we talked about the Taylorac brain versus those Montreal Neurological Institute or MNI brains. We talked about the advantages to each and the disadvantages to each. Talked about the disadvantages mainly of Taylorac. So now people tend to use this MNI brain most frequently. Okay, so that's our review of last week. I think if you want, we can take a little break now, and then we'll go on to talk about fMRI data modeling when we get back. So if anybody has any questions, please let me know. Okay, so we're going to only take a 15-minute break today because we are going to go an extra hour because of the holiday yesterday. So I'll see you all in 15 minutes. Okay, so we're going to go on to talk about modeling the fMRI data at the first level. Okay, so there's two distinctions in modeling fMRI data. The first is first level analysis. So first level analysis is when you take an individual's data and do the statistical comparisons and modeling to find where for that specific person their activation was. So that's, again, first level analysis is for the individual subjects. Second level analysis is when we take that first level analysis and we combine it with other participants to get group analysis. And we will actually talk about that a little bit later in this lecture as well. Okay, so just going through our basic outline of fMRI, we talked about how it works. You know, this went 1, 2, 1. Again, I try to fix that so many times. PowerPoint likes to do its own thing. So it should be 3, 4, 5, 6 anyways. So we're on point number 4, how to analyze the data. And again, here's where we are. We've talked about pre-processing, normalization into the common space. Now we're going to talk about statistical analysis, the general linear model, and how we get T-maps, F-maps, and do some time series analysis. Okay, so the first question I want to pose here is, what constitutes brain activation? So how do we know what areas of the brain are active compared to other areas when a person alternates between two different tasks? So we use statistics to help us answer the question of what constitutes activation. So how do we determine whether an area is active or not? How confident are we that what we find is activation? How are the results in a group of subjects applicable to a wider population? And are the differences between groups of subjects significant? So again, the first level is taking our individual subjects and analyzing their individual data to find their activation, which is a statistical technique in itself. And then when we combine these groups of people, it's again another statistical problem. But they all sort of ask the same question. So how do we know what a difference is in an activation or across a group? So if you guys are familiar with basic statistics, you've probably seen this Gaussian curve. Okay, so the Gaussian curve takes the mean of the population, which would be right in the center, and then this is a histogram showing as the population varies, you get the sides of the curve. So if the person's or whatever data you have fits right in the middle there, then you assume it's statistically like other data. Okay, so once you start getting in these outer areas of this Gaussian curve, then you can start making inferences that this 2% over here is probably different from others, and on the other side as well is probably different from others at the outer parts of this curve. So what we're going to do to find brain activation is fit our general linear model, and we're going to use our estimated model parameters to see if they're the same as others or less or more than others. And we're looking for differences that fit in these little outer reaches of this curve so that we can say it's different from something else, then we can call it activation. So once it exceeds a certain threshold here, then we can say we know this is activation. And we'll go through this in more detail. So we're assuming in event related or block, depending on our timing, that we have this turning on and off of a signal. So if we have our task, we can say, okay, so the lower part of this blocks are at rest, the higher part of this blocks are when this person was active in a task. And we're going to try to fit this curve to it of whether our model looks like what's changing in our model looks like what's changing in our recorded data. So we're going to try to fit our model to our data and estimate parameters to see how well it fits. Okay. So to do this, we're going to apply what's called the general linear model. So what we have is in fMRI data is Y. Y is our observed data here on the left side of the equation. Okay, X is going to be our predictors. So this is going to be how we design our task, whether our person is at rest or whether our person is in an active state. And we're going to try to fit this using parameters, which we're going to call the beta weights. And then there's going to be an error based on how well or how well it fits or does not fit our data. So again, our observed data is the bold signal. This X is our design matrix, and we're trying to estimate these beta weights, and we're going to have an error. We're going to talk about this a little bit more. This kind of visually shows it. So if our observed signal looks like this vertical signal on the left, our predictor is this block of turning on and off, and we're going to use this beta to tell us how well our observed data fits to our predicted data. Okay, so I showed you in the case of when we had one predictor. So we could add second predictors, and people do. It depends on how complicated you want to get your model and how well of a fit. You can add as many parameters as you want. So another parameter that you might add, or somebody might add, would be the first derivative. So this is just an example of how we might say, okay, our observed data Y is now a function of our model, which would be that first X1, our turning on and off of the task. And we could add, again, another predictor. Maybe the first derivative, in this case, might allow for time shifts or variations in our design. And so we could add this as another parameter to try to predict our data. And then in this case, we would get 2 beta weights for our model. So I said Y is our observed data. So what we mean when we talk about this observed data is a matrix of individual voxels. So in this case, we would take a single voxel in our data and track this over time. So we might take one slice of the big brain, just pull out one of those little squares that we looked at, and track that over time to see if it changes according to how our model is changing. Okay, see how well the changes in that data fit to the changes in our model, or the turning on and off again of our task. So again, we're doing this for each voxel individually, a single voxel, looking at how it changes over time. And you could represent these, instead of a time series like this, by intensity changes in a grayscale, because that's actually what we're looking at. We're looking at how the grays and the whites vary in this signal. You could represent one voxel in terms of a column here, you see within that red. And so instead of looking at the peaks and the valleys, we're looking at the changes of the gray intensity over time. Okay, so I took some of these slides from Elliot Freeman's Idiot's Guide to the General Linear Model, fMRI. And so he wanted to point out here that you could model any type of moving data with this over time. So if you had a predictor of how you expected the signal to vary over time, you could even take an amoeba that's wiggling around in space over time, and you could do this same type of modeling on it. So it's not specific to our brain and what's happening in the brain. This is a model that can be applied to anything observed over time. And we use the software called statistical parametric modeling as the way that we do this modeling. It's called SPM. This is a commonly used statistical package for fMRI data. There are also some other types of packages available which do similar things. So it really is sort of a preference on what type of software you like to use. People generally use the type of software that they were trained on, and hopefully they are adaptable if they want to use another program to see if they get the same results. So that's always interesting to compare software programs for the same data. But what the things that I'm going to be showing you here are taken from SPM, so statistical parametric modeling, and that's what we use in our lab to model this data. Okay, so let's get a little bit of idea of how we're going to try to fit our data to the model. Yes?
So basically you just want to consider how well a particular voxel is fit to your model to determine if it's part of the activity.
Correct. That's correct.
But then how do you design the initial model?
So we're going to design the model as using this assumption. It's a linear fit, so it's a one-to-one. Basically, when we're in the case of a linear model, you're assuming when your task is changing, what you're seeing in the data is going to change accordingly. So it's gonna, if our task alternates between rest and activity, then we're assuming our data is going to change accordingly. So our data is gonna alternate between a certain place at rest and then a certain place while it's active. And we're gonna assume this is a linear fit. Yes, the best fit line. So I'm gonna show you a little bit more in the detail here, and maybe it'll help to understand it. Okay, so say that we're going to look at how difficult our task is and vary this from a number from 1 to 6. So 1 means our task wasn't very, well, I guess it's 1 to 7 on the plot here. So 0 to 1 means that our task wasn't very active, and difficult, and 6 means that our task was very difficult. And we're going to take our observed data, which is y here, and we could make a plot like this, okay? So we could make a plot of y and x. So in the first case, if our task was at a difficulty level of 1, we would plot the 2 points that we see corresponding to the 1 on this chart here, okay? So this would go this point, and we could plot them here, okay? So as it goes to 2, we would take where our task was a difficulty 2 and put them here, okay? So similarly for 3, 4, 5, and 6, and then when we do that, we're assuming that as our task becomes more difficult, for example, we may say our intensity that we see in the image is going to increase, okay? So we try to fit what we see in the data to how difficult the task was. And we're assuming this is a one-to-one relationship. So if our task is more difficult, our data is going to change accordingly. This is a linear fit, okay? So if we were assuming, you could make other assumptions of nonlinearity. So you could assume that maybe it would fit in with a quadratic equation, right? So you could assume as your task got more difficult, the data might fit in a different type of way. This would be a nonlinear fit. But when we try to fit it with a straight line through the data, that's a linear fit. So even if our task looks like a nonlinear function going up and down like this, our bold signal, right? We're trying to model our data fitting onto this bold signal. So it's, again, it's a one-to-one relationship. If our task difficulty goes up, our signal goes up. If the task difficulty goes down, our signal goes down. Does that make sense? Okay. So we try to fit our data like this in a linear way. And we can calculate what is the best fit of this line. And you do this using a least squares estimate, okay? So the specifics of the least squares estimate isn't shown here, but you can see here that when you try to fit your line here, you try to minimize how far each of those points lies from your line. And you can do this mathematically and to get your best fit line through there. So the slope of this line is what our beta parameter is going to be, okay? And if you guys know linear equations, y equals mx plus b, you're going to have the slope, and you're going to have where it's going to cross the y-axis, right? So we're going to especially try to look at these beta parameters or our slopes to see how well or how much change occurs when our signal change. So if it's a steeper slope, then our signal is changing more with the data. And, well, really it depends on the model and the fit, okay? So how well does our model fit our data in a linear way? And again, these little deviations from the line are what makes our error terms. So this slope, how we process our data. So we're not so much interested in what the numbers are. We're interested more in how they're changing with our task, okay? So if you see something like 57 or 56, we may not really be able to say, okay, that means there's activity of 57. Instead, we're trying to fit this because these intensity values could be normalized or they're relative to the whole image over time, okay?
How is the task difficulty computed?
So this would be just one example. And so the experimenter would set how difficult the task is. So they might say, okay, at this time point, we made the task this difficult, and at this time point we made the task easier so that we could keep track. And it really depends on what type of task it is and what they're doing. So you would have to assign that ahead of time. You would have to make a determination ahead of time of task difficulty and how that changes. So again, since we're doing this linear model, we have a constant, and in this case, that's where it crosses the y-axis with our fit and our beta estimates, and that's how well our task fits to our observed data, okay? I know it's a difficult concept to conceptualize at first, and especially difficult if we start adding more modeling parameters. So we could continue to add all kinds of modeling parameters if we wanted to. So we could say task difficulty, we could say maybe the first derivative, we could, and then you'll see we're even in a minute here, we're gonna add motion estimates as part of the model so that we can add motion in to try to explain how these intensity values are changing. So each time we add another estimator or another X, we're assuming that's contributing to how our intensity changes are changing over time. So we can say these intensity changes are only due to our task going up and down, or we can say they're due to the task going up and down, plus the person's motion as they're moving around. So we can try to we can put different things into our model to try to explain what's making our data vary over time. Okay, so again, once we define our model, then we need to estimate the parameters of our model. Again, finding these beta weights, okay? So we do this again with the least squares approximation, and luckily our software does this for us. Because if you imagine trying to do this for, say we have 64 by 64 voxels in one slice, and then we have maybe 30 slices, we end up with thousands of voxels. So in order to do a least squares fit of each of these, that's a big computation. So we're going to estimate these model parameters. Then we're going to use these model parameters to try to see if we have activation. We're going to use these betas that we can compute to try to compare different parts over time to see if we have activation. Okay, so again, we can have these. So I'm going to try to explain the difference between covariates versus conditions. So our covariates are, so these are what we're trying to see if they're affecting the changes of the independent variable, which is our bold or our Y value. So we're looking at covariates of the model to see if these help us understand what is going on in our data. Conditions, on the other hand, are where we specify these are happening. So is it on or off? Are we in rest or are we in an active state? Okay. So we use these and we put these together to try to understand what we're seeing in our data. Okay, so in SPM, you get basically, it represents your model like this, okay? So you see all these columns here. So this would be also, okay, so this also includes motion. So like I said, you can also use motion as covariates in your model to try to explain what's happening in your data. So I'm going to go over to the board here and try to explain to you what each of these is, okay? You would see a line, sort of the squiggly line of how the person's moving, but you can also represent this in intensities rather than in numerical values. So they're still numerical values, but they're being displayed as intensities here so that we can see them in our model. So it's okay if you don't understand this completely. I just wanted to show you how SPM represents this, and we'll go through a very specific example next. So that might help you when you see that as well, and looking at actual conditions of the task. Okay, so we call this modeling our hemodynamic response function. And so I told you that we're going to do this on and off with our task, okay? So here's our task going on and off down here, and those are our scans. So we're assuming that our scans are going to change with our task. Our intensity is going to change with our task. But what we have to remember here is this hemodynamic response function is a slow function, okay? So when we do the task, we are not going to assume that the brain is instantaneously changing. We remember that the bold signal takes about 6 seconds to peak, and so it has a little bit of delay before it turns on. And so we're going to use that when we model our data. We're going to allow for a little bit of delay between when we have our task and when we assume that we're going to see our function. Okay, so we're still doing this linear design. We're still trying to take a function and laying our data on it and see how it changes with the task, but we're going to add a couple seconds, usually about 2 seconds delay in there, so we can account for the slowness of our bold response. Okay, so basically to summarize what we're modeling here, we're starting with our finished product. That's our observed data. And we're trying to add the ingredients. That's our design matrix when a person is doing a different thing. And as we're doing this, we're trying to find how much of each ingredient makes what we see in our finished product. So it's just a sort of just a conceptual thing. And then we can compare what we estimate in this model to the different parts of the task, so we can see if we have activation during the task. Okay? So basically, again, we're starting with our output, our observed data. We know how we expect our data to change based on those X's, and we're trying to see how well each of those observed voxels changes with our expected changes. Okay, so back to this question now. Once we estimate these parameters, what do we do with them? How do we know whether an area of the brain is activated or not? And again, we're going to use statistics to look at these differences, okay? So we're going to use what's called this hypothesis testing. And if you've taken statistics, you've probably seen this already. So our research is going to be framed as a hypothesis. So the null hypothesis assumes that it's, or whatever's happening is not true. Or, so here might be our questions that we're going to ask, okay? That condition 1 equals condition 2. So the brain during, or the voxel during looking at a happy face is equal to the brain looking at a neutral face. And we're going to say that is the hypothesis that we're interested in, H1, okay? Our null hypothesis is that there is no difference in a voxel when we're looking at these, okay? So we're trying to test, is there an actual difference in these? We're going to be testing our beta values for these, our model fits. Are these beta values the same when we're looking at two different conditions, or in two different conditions, or are they not the same? Okay, so here's our hypothesis. Moving the thumb will cause an increase in neuronal activity, which we detect with the bold signal changes. So again, let's just think about this as a single voxel over time, okay? And we want to compare this voxel as we're moving our thumb and as we're not moving our thumb to see if there's a difference between these two states. So our null hypothesis in this case would be moving the thumb will not cause an increase in the neuronal activity. So again, our experimental condition now, we have two conditions, moving our thumb, not moving our thumb, and so we have two X's. We're going to estimate the beta for this voxel while the thumb is moving, and the beta while the thumb is not moving. That's our control condition. We're going to say, is our experimental condition equal to our control condition, or is it not? And we're going to use this fMRI data over time to answer this question. Okay, so this is just some basic statistics here with hypothesis testing. So we could have a distribution over time, and this is just for conceptually. Say so this is a mean and a standard deviation of our intensities as the thumb is moving in the green, and as the thumb is not moving in the purple, okay? So our distributions of the data could look like this with no overlap. Okay, they could look like this with quite a bit of overlap. Okay, they could look like this. So maybe instead of coming to a peak in the number of scans, they would be more widely distributed and lower. Okay. again, here we could have something that looks like this as well. So we're looking at our distribution of the number of scans and how they're, the mean and the standard deviation of the intensity is as we try to compare these two states. Okay, so there's two factors in trying to determine how much the effect that the experiment had on what we see. Okay, this could be the differences in the means. This is the peaks or the means, so we could see how much difference there is in those and how much degree of overlap are between those curves. So let's look at this again. If we saw our data looking like this, we might conclude that the experimental condition, that is moving our thumb, had an effect, so that we are seeing differences in the means, and there's not a lot of overlap between these in either of these cases. Okay, so in this case, we would conclude that our experimental condition had an effect. But what if we saw something like this? Okay, the means are very close together, and there's a lot of overlap between those. In this case, we would conclude that our experimental condition had no effect. Okay, so how do we know really if these are different distributions or not? So thankfully, statisticians have spent a lot of time thinking about this, and the t-test is what formally incorporates our intuitive sense of if there's an effect or not by calculating a measure of how different or how similar these distributions are. And so this is a t-test here. What it is the mean of the first distribution minus the mean of the second distribution over the square root of the sum of the standard deviations squared. So you can see the equation here. This is just a very common statistical technique that's used to test two different hypotheses. And so now we might use our beta values in these instead of the mean. So we want to test if our beta values in two conditions are actually different or not. Okay, so once we get these t statistics or these t equation measures, we then we can look at a table saying if this is a normally distributed measure, then we can look up to see whether based on how many the number that we had of these, see how the probability that this event would occur randomly or not, see how much that is true. Okay, so we can take our t measure and convert this to a probability measure and say this is the chance that this t value shows that this event would occur randomly or not. So this actually can give us a numerical measure of how confident we are that there is a difference between these two distributions. Okay, so this is our p measure. So in science, these p-values are sort of the gold standard, okay? So you want to select a p-value. You might select a p-value, p is less than 0.05. Okay, so the smaller the p-value, the more significant your experiment is. Nowadays, and this p-value again is going to depend on this t statistic that you calculated and what's called the degree of freedom. So the degree of freedom is actually the number of, based on the number of samples that you have. So in a single subject analysis, the degree of freedom would be the number of total scans you have minus 1. Now, there's been a lot of arguments over this p-value lately in the field of statistics, and people saying that this p-value is given so much weight in studies, especially studies of medical experiments or research in medical applications, because sometimes these people don't have a really good idea of what these numbers actually mean. So probably me included, I have my basic idea of it, but the statisticians are all coming back and saying these p-values aren't good enough. So there has to be other ways of trying to determine if a result is real or not based on these p-values. So that's just something that statisticians that I work with have been focusing a lot on lately. How do we actually know if something is different or the same? And things that might show us if they're different or the same would be reproducibility of a study. If the study could be reproduced across labs and they would get the same results, and types of things like this. Okay, so after we compute this t-test, here's how we might determine how significant something is. So this is our t-distribution. You can see it goes from 0 and it goes to high numbers over here. High numbers being maybe greater than 2.5. And it goes to lower numbers at the end at the other end. So those are the negative numbers, and our t of 0 is right in the middle. So we could look at a one-tailed t-test by only looking at one of the sides of this t-test. In this case, we would make an assumption over whether one distribution was greater than the other, and we would only test to say, condition 1, when thumb tapping, we expect this to have greater activation than condition 2. So we would limit it to a single side or a one tail of our t-test. But say that we want to say, okay, maybe this isn't true. Maybe our intensity values are actually less when person is tapping their thumb over when they're still. In this case, we would have to look at both tails of our t-test, okay? So in this case, we could assume that thumb tapping may cause an increase in activation or a decrease in activation. So we'd have to look at a two-tailed t-test. Okay, so again, back to this t-statistics and p-values. Suppose we find a voxel in which the t-statistic is 3.3, and there were 20 scans that we took at rest and 20 scans that we took while moving the thumb. So the probability that the difference in the MRI signal in this voxel is not due to the movement of the thumb is 0.002 for a two-tailed or 0.001 for a one-tailed. So this would be, again, we can use tables to convert these t-values to these p-values. Somebody's already nicely computed all of these for us. And nowadays, though, you can go online and find all kinds of statistical calculators. You just plug in your numbers and it'll give you a p-value for this. Okay, so how does variance in our data, or how much of that data doesn't line up to our mean? How widespread is that function? How does that affect our data? So is it better to have a lot of variance or a little variance? And this kind of shows just an example of that. The data set on the top might be a voxel that has a lot of variance over time. So it's down low, then it's up high, and then it's down low. You see there's big separation between our peaks and our valleys here. And in this case, we would get, doing all the calculations, we get a t-value here of 2.3. If we have less variance here on the bottom, we see less spread. We see less spread of this distribution. And we actually get a much higher t-value for this case. So variance, an increased variance, gives us more uncertainty in the result that we see. So if there's less variance and a stronger mean, we're more certain of what our result looks like. Okay, so unlike the t-statistic, the p-value for z-scores which are based on the normal distribution and doesn't change depending on the number of scans. So sometimes people will take their t-value and convert it to a z-value. A z-value is a standard normal distribution. That's with it's with the mean of 0, right, and a standard deviation of 1. So you can take your data and you can normalize this into the z-score, and then it's more it's easier if you use a z-score to compare across different studies rather than constraining to this t-score. Okay, so problems with the t-test. Systematic differences such as artifacts can create apparent significant differences where none actually exist. So again, we could have motion in our data, and this motion's causing our intensity values to change over time, not because the tissue was active, but because as the person moves, we're seeing different pieces of tissue over time. So the intensity is changing because we're looking at a different structure. So think of this on the edge of the brain especially. So you're going from a lighter intensity region of the brain to a dark area where there's no tissue. So say that border is fluctuating back and forth as a person does the experiment. So especially if the person is moving in accordance with the task, and we see this happen sometimes. So person might be tapping their thumb, and while they're doing this, their head might just be moving very slightly because of this movement that they're doing in their hand. And it's hard to imagine that the head could change the emotion that much from doing something on the extremities of the body, but it's true. And sometimes some people have more of this than others, or they're say they're excited when they see something. So every time they're pushing the button, their head is moving a little bit. This makes the motion correlated with our task. So the brain voxel might be changing due to this slight motion rather than actually changing due to our bold effect. This is going to give us artifacts in our data, which is going to give us false activations. So to decrease this effect, 1, we can do motion correction, and 2, we can add those motion parameters into our model to try to account for these differences that are occurring due to the motion rather than the bold effect. Okay, so the t-test also disregards any time characteristics of the data since only the means are compared. And the assumption of the t-test is that the data is normally distributed. And this is not always true. So this normally distributed means this Gaussian curve, those nice smooth curves that I showed you. And that assumes there's a mean right in the middle and that the variance is the same on the positive side as it is on the negative side of the mean. So this is assuming that all of our data has this nice Gaussian distribution. And that is not always the case. That is one advantage of spatial smoothing. Spatial smoothing helps our data to become more Gaussian shaped. So it helps the assumptions that we make in our underlying statistics to be more true and more accurate in our results. Okay, so there's another problem of doing this, okay? The problem is that we might have 64 voxels by 64 voxels over 30 slices of data. So we're actually doing thousands of t-tests, okay? And we're assuming that each voxel is working independently of the others. So when you do thousands of t-tests, what's the chance that you'll get activation just due to random chance, right? We're doing all these t-tests and we're saying we're going to make assumptions that if it changes with our experiment, it's active. But if you do t-tests over and over and over again, people have shown that just randomly, sometimes you will get what's called these activation areas. So there's called type 1 error and a type 2 error. And this is also thought about in a lot of other types of medical sciences. And so let's pretend instead of this voxel that we're testing somebody for a disease, okay? So we're testing thousands and thousands of people for this disease. So if we tend to do a lot of experiment testing, we can start seeing these false positives and false negatives in the data. So we can divide our experiment into, or our case of this disease testing into, they have the disease or the condition, and they actually don't have the disease or the condition. So this is the actual truth here. They have the disease, or they don't actually have the disease. So they do the test for the disease, and the test can come back either positive or negative. And that's the column here on the left, showing in the rows. They actually test positive or they test negative for the disease, okay? So if they have the disease and they test positive for the disease, that's box A. That's called a true positive. If they don't have the disease but they test positive for it, it's called a false positive. That's square B here. So say they have the disease but they test negative for it, that's called the false negative. That's here on C. And say that they test negative but they actually don't have the disease. That's called a true negative. So that's box D here. So the problem comes if we get box B, a false positive. In this case, you would tell the person that they have the disease when they actually don't. And then similarly to ours, we would see and assume that a voxel is actually correlated with the task or active when it's not. Or the other problem would be is if they have the disease but they test negative for it. So this was this false negative. And this would be when we don't see activation but the voxel is actually active, okay? So this is a problem called multiple comparison testing problem or multiple comparison problem. This is when we do thousands and thousands of t-tests. Our chance of making these types of errors is greater and greater due to random events. So the more tests that we perform, the greater the likelihood of making errors. And so in this example of 64 by 64 voxels in the XY plane, and we have 32 slices. So if we do this calculation, we actually end up with 131,000 comparison tests. So this becomes a problem. So which voxels do we see are actually significant, and what voxels are coming up that we see that are positive when they're really positive or active when they're really positive, and not active when they're really not active. So sensitivity is called our rate of true positives, and specificity is our rate of true negatives. So I know we've gone for an hour, so maybe we should take a little break, 15 minutes, and then when we come back, we can talk about this multiple comparison problem some more. Okay, everybody ready to start again? Back to our multiple comparison testing problems. Okay, so again, just a reminder, so if we're looking at 64 by 64 voxels, we're talking about in an XY slice. So 64 divided 64 ways this way, 64 ways this way, and 32 slices throughout the brain. So for each of these of the whole brain, we're recording over time. So every single voxel has a time series. So say we do 5-minute study, so we can track each of these voxels over the 5 minutes and do these comparison testings to see if we have activation in brain regions or no activation. And again, the problem is that when you do as many t-tests as this, in this case over 131,000 tests we're going to randomly get activation that is not really attributed to what's going on in the brain. So this could be a false positive if we see activation where there is none. And we'll also see no activation when there is actually activation. So we could have a false negative or a false positive. And if you study statistics, you see that sensitivity is the rate of the true positive. So how many times are we doing a test where it's actually should be true or a positive result and it's actually true? This is how sensitive our test is. Or how many times are we getting a negative result when it's a true negative? And this is called our rate of specificity, how specific our test is. Okay, so there's ways to measure and quantify the amount of false positive, and by using these different things, there are ways to also control these false positives. So there's two different tests that I'm just going to go through briefly. One's called a family-wise error rate, or family-wise error testing, and the other is called false discovery rate. And the family-wise error is going to control the probability of false positives in our data. And our false discovery rate is going to control the proportion of false positives among the tests that we reject. And I'm going to show you just really briefly how these work. These are two methods that people use in fMRI to try to control these types of errors. So the family-wise error rate controls the probability of making one or more type 1 errors. So again, type 1 error is a false positive. So you're seeing a positive activation when there actually is none. And to do this, we adjust a p-value for the number of hypothesis tests that we're doing. So we take this p-value and we use it to adjust based on how many t-tests that we're doing. And the way there's different types of methods to control this family-wise error, including Bonferroni correction, random field theory, and permutation tests. Okay, so how do we avoid making false associations or false positives? So say we're doing M independent statistical tests at a significance level of A, okay? So M might be 131,000. That's how many t-tests that we've done, because that's how many voxels that we're tracking over time. And we pick a significant or a p-value of 0.05. So we want to actually know how we want to control for the significance A. So what we want to do is we want to take A, which is this rate of control, and we want to calculate it based on our p-value and the number of tests that we use. And so they came up with this formula to do this. And so we can therefore say A is estimated with this adjustment. So we would take our p-value of 0.05 and we would divide it by the number of independent tests that we did. So in this case, this is called Bonferroni correction. This was developed by Carlo Bonferroni in 1935. So he says our p-value should be adjusted by taking our p-value that we would choose. So for this case, 0.05, and divide it by our number of statistical tests. Okay, are you guys all writing? Did you get that? The problem with Bonferroni, so there's good things and bad things about Bonferroni. For one, it's a very simple method for ensuring that the overall type 1 error rate of our p-value alpha is maintained when we perform multiple independent tests. So it rejects any p-value that's less than or equal to our initial p-value alpha, so 0.05, divided by the number of independent tests we have, 131,000. So if we have an experiment of, again, 0.05, and say we only do 10,000 voxel tests, then we would need a p-value of 5 times 10 to the minus 6 to get any kind of significance out of this test. Now, the problem with this is extremely conservative. So anybody who's worked in statistics knows p-value of 5 to the 10 to the minus 6 is pretty conservative. So the problem with this is that it often corrects to the point that we see very little activation. So this is an example that I found that looks at what our activation might look like. At first, on the left, a p-value of 0.05. So p-value of 0.05 in fMRI, we consider that a fairly liberal p-value. So we're seeing a lot of activation. This, I'm not sure what this task was, but the putamen is activated and the dorsal ACC. So we see this a lot in our studies too. So this could be something of fear or some kind of emotion activating these areas. Now say we reduce our threshold by making a more strict p-value of 0.001. So you see how much our t has to go up to get to this p-value of 0.001. This is not a huge difference, and we see less activation when we constrain our p-value. So you see a lot of the stuff around the border of the head up on the left and the right here has gone away when we increase our threshold. So we're going for a more conservative threshold. Let's say we want to use this Bonferroni corrected technique though. You can see that now we have to have a t-value from our original data of 7.15. That's a very high t-value. And what we've done here at p of 0.05 after Bonferroni correction, so again, we would take our 0.05, divide that by the number of tests that we've done. That gets us to this high t-value, and you can see how much activation is gone now after we've done this Bonferroni correction. So it's a very conservative technique to do multiple comparison tests. In 1995, Benjamini and Hochberg developed this false discovery rate type of test. Now this is also used frequently in fMRI. So when we do this, we order the test according to p-values. So say we can take for a specific t, we might look at our p-values, or we could pick a p-value. And what we do is we limit, okay, so we would order the p-test and we would limit only at a certain point, we would cut it off. So for false discovery rate on an alpha of 0.05, we might find that this J star for significance level of J going from 1 to J star based on our p-values, then we would pick a point where we cut this off. So we assume that J greater than J star are no longer significant. So we would order results according to our p-values. We could define p-values in order. Then we can say after a certain control point, we cut this off, and any p-values that are less than this are no longer significant. So this is more of a way to sort of order our significance and then cut off at a certain point, rather than taking something and dividing it by the number of tests we do. So this technique is more based on our preferences of where we want to limit the activation. Okay, so here is family-wise error versus false discovery rate for working memory. And okay, so on our false discovery rate, if we use this limitation, we see that we get this high t threshold of 7.67. We have very little activation there. We see that only 58 voxels now are active in this whole brain experiment, and based on those 131,000. Whereas if we use the same technique with our false discovery rate threshold, and we stopped at a P or so the T and the P values are sort of similar because we have the same number of experiments, so we can take P to T and T to P back for a fixed number of scans or a fixed degree of freedom. So it's also like we order our T values and we say anything below a certain adjusted point we're not going to consider active anymore. So we're making an adjustment and we're thresholding at this particular place where we say after this we won't see any more activity. So if we use this false discovery rate test here, we see that we get more activation. So it's a much less conservative technique, or we are actually letting more activation through by using this false discovery rate correction. Okay, so once we determine our activation regions, how do we actually visualize where there's activity in the brain? So the output from analysis software is typically going to include some visual representation on the brain and some tables of activation. So there are a variety of number of software tools that are available to integrate and visualize these results together. So we're going to talk about how we would do this. One is reporting a local maxima. So say we find a cluster of activation in a particular area of the brain. How we would report this cluster is by finding the peak T value within that cluster, reporting that peak T value and the coordinates of that T value. So we could also define how many voxels are active around that cluster. So at a certain threshold, we can find a cluster. We can say this has a peak T value of maybe 4.5, and there are at a P value of 0.001, we see maybe 30 voxels that are active around this peak T value. So that's one way that we could report this. Now, when I say clusters of voxels, the criteria is that all these voxels have to be spatially connected to define a cluster. So we say we cut off at P equals or less than or equal to 0.01. So then we might see a cluster of voxels that are all together and active. So we'd say that this is an active region. So they all have to touch each other to be considered contiguous voxels that are activated. So in SPM, this can it'll do this automatically for you, and if the voxels are 8 millimeters apart between peak points, then it can divide them into two different clusters. So we're going to see this a little bit more as we go along. Another way to look at our results is to just take a look at the time series of a voxel and see if it actually is changing according to the characteristics of the on and off of our task. So this would be sort of a time series analysis way to do this. Or we could take a group of our active voxels and we could average the time series of these all together and try to look at how well the pattern of these intensity, brightnesses, and darknesses follows the pattern of the on and off of our task. So that might be another way of how we would look at these. So when we go to display our results, what we typically do is we don't leave them in this grayscale. We add color. So we define colors associated with different T values, and then we can show where the most active regions are in our brain. So in this case, the areas of yellow would be the highest activation. You can see on the left there that they appear whiter in our intensity image. So in order to make these stand out on the background of the brain, again, we might assign color values to them and then include a color scale map in our results. which is not done here. And also, this also looks like a lot on the border, so that looks like it could possibly be some motion artifact there in our data. So it's hard to know without actually looking at our motion estimates. So anyways, we use this to get a basic idea of where we're seeing activation in our brain maps. So another thing that's used in SPM and also some other software programs is what's called the glass brain. So the glass brain is a quick way to see where there's activation. And what this shows is the 3 slices of the brain. So at the top left, we have the sagittal view. Top right, we have the coronal view, and the bottom left here, we have the axial view. And these all show the same dots on the different views. So all show the same activation points on the same view. And these are the 3 orthogonal planes again, the sagittal, coronal, and axial. The thing about these glass brains is it's kind of hard without looking at these a lot to actually get an idea of what you're looking at. So you see where this little, can you see that little red cursor mark in this brain here? But it shows this spot of activity. So when you look in the coronal view, this is where you are with that spot. And then when you look at the axial view, this is where you are. So we're looking at this spot in each of the 3 planes. So they each give a unique sense of where this spot is on the brain. So you sort of have to incorporate these 3 views together into your own brain and integrate your sensory information to understand where this spot is. So when I started using fMRI, these were a little bit confusing to me, but I sort of get a more general idea now. You can see that a lot of the activation due to whatever task they were doing here is more in the mid to front range of the brain, and they seem to be more here on the right side, and on the axial view again, you can see that it's more towards the front right of the brain. Does that make sense to everybody? And of course, we want to fuse the functional and anatomical images together. So this is another way to display our activation. So again, we've co-registered these 2 brains. We've lined them up for a single subject, and then we've overlaid them onto the anatomical space. So we can actually see where in the anatomical brain tissue our activation was attributed to. And once you've warped your brain into this MNI space, SPM has this standard underlay that you can use to display your results on. And it looks much prettier when you display them on an anatomical brain rather than the functional brains. All right, so what are we going to do with this activation information? So we've gone through all these complex computations and came up with these statistical tests, and we've said, okay, now we see where there's activity in the brain. So what are we going to actually do with this information, right? Okay, so one thing we can do is this called whole brain analysis. This type of analysis is more exploratory. So say you want to just know what regions of the brain a task is going to activate. We would do this whole brain analysis to look individually at where the brain is activating for each subject. Then we could put these together statistically to make these group maps and compare different groups of subjects. So we can look at different groups of subjects and say, are they using approximately the same areas of the brain to do the same task? Another thing that we can do is this region of interest analysis. So instead of looking at the brain as a whole part, we start breaking down the brain into the little more of the anatomical features. For example, we may focus on the area of the amygdala if we're doing an emotional task. And we could apply a threshold, and then we could say, for different people, how many voxels in the amygdala were active compared to the total number of voxels in the amygdala. So we could also look at, for example, we could do a correlation analysis. So depending on somebody's IQ, we could say what percentage of their voxels in a specific area were active compared to non-active voxels in that region. So we could use this to look also at differences across different subject types as other things varied, for example, their IQ. So in a region of interest analysis, we could define regions by anatomical features, or we could also divide them down into other types of regions. And like I mentioned at the beginning of the class, one region, regions that are typically used today are called these Brodmann's areas. This was, Mr. Brodmann took all the different tissue types. He took a brain and he dissected it, looked at different tissue types, and gave them each part a number that is associated with a different function in the brain. Now we see that a lot of the same numbers or adjacent areas are responsible for example, for vision. So a lot of the blue areas here in the back of the brain that you see are different Brodmann divisions, but they're all responsible for the function of vision. Somatosensory, again, is the somatosensory strip to here. So we can divide them according to Mr. Brodmann's divisions. And since these are standard universally, people will know what area you're looking at based on the Brodmann number that you use. So the last thing that we're going to do with this data is create these group maps. Now we're at second level statistical analysis. So again, the first level analysis focused on just the individual's brain and finding regions of activity within a single person's brain. Now we're going to take the statistical things that we calculated for each individual and we're going to put them together. So let's see how we're going to do that. Whoops, I need to do that. Okay, so there's some problems of doing these group maps. One is that fMRI experiments and MRI experiments are typically very expensive. So I think I mentioned before, it's cost about $600 an hour to run an experiment. This is at a contracted research rate. So this is for at the research center. So a clinical scan may actually, they may charge a lot more for this. So this is the rate given by like our facility for people with grants, so that when you're writing a grant and you're making a budget, you can say, I want to look at 100 people. So you need to budget in your into your study how much that's going to cost for each MRI scan. And you can see that it quickly gets very expensive to scan people. So that's one problem, that you typically can't do maybe like 1,000 people or something. It's not like doing some other type of statistical collection where you may go door to door and give surveys to people. It's an expensive and it's a very time-consuming technique. All the brains, again, have to be warped into this normalized brain space. So you're taking everybody's single brain and you're kind of distorting it and moving it around. So this isn't really ideal, like I mentioned before, but it is something that people use to get an idea of group activations. So we need a method for pooling this statistical data. So we're taking statistical data and we're going to do more statistics on it. So this isn't really an ideal thing to do, but it's what's typically done. And another issue is that levels of activation vary across subjects. One problem of studying human subjects is there's large variation between subject and subject to subject. So these can be accountable for a lot of different things. It could be somebody's genetics, it could be what they ate for breakfast, it could be maybe medications that they took that increase the blood vessels, maybe an aspirin or something like that. They could be on iron pills, which might change the bold signal. So these are things that can vary across people. And some of these things we can control, and some of these things we can't control. Okay, so we can look at activation for a single subject. So then what do we want to compare these two subjects? So again, we need more subjects than two if we're going to get statistical power, right? So say we want to look at some kind of difference in their visual cortex back there. If we just look at two people, we don't have a lot of power to make any conclusions. So again, we're going to normalize into this brain space by lining up landmarks and by warping the shape onto these Taylor-Rack atlases or the MNI templates. And although this is a standard procedure that's used, again, it introduces spatial smoothing when you start resampling data, and it also introduces other errors. So it's not always the ideal way to do it. Again, you might want to take a brain and put it into Taylor-Rack space. So you might have this before and after sort of warping of the brain. And you're moving this tissue around again, and you're resampling it. And this changes the data to some degree. So this was from my study. So I was just looking to see how much variability across the years. So this would be, the top row here is the same person. It's the same task with two different people. Okay, so you can see how much variation there is. The one person had a large areas of activation. The other person hardly had any for the same task. And so this person at the top left, I scanned 1 year, and they came back the next year, and I scanned them again. And these are the differences that I saw using the exact same task on the exact same subject at different time points. So you can see you really can get a lot of variation in this data. So what would be the ideal way to statistically combine this information? So again, we're taking individual results that are already based on statistical testing, and we're doing some more statistical testing. So there's a number of different techniques that people use to combine statistical information. And these can be one of two categories. One is to try to combine the statistical outputs from each of these subjects into another statistical test. And these types of combining techniques use t-values and p-values from individual subjects to compute a group result. The other is called meta-analysis, which uses fixed and random effects models. And these actually go back to each subject and use the means and the standard deviations from each individual subject to try to combine these. So you could either go from a range of something pretty easy to a range of something pretty complex when you try to combine information. So the top row here just shows some of the different techniques that can be used to compute the statistical information, to combine it. These could include Fisher's method, where you're using this computation of the p-values. You could use, again, Stouffer's method, where you're combining p-values, or you could even use an averaging of the t-maps across subjects, right? So that you would just sum everybody's t-values divided by the number of people that you're looking at. Or again, you could do something more complex like the random effects model, where you're going back to your original data for everybody and trying to combine in this way. And the random effects is probably a better model to use in this case, but it's very computationally intensive. So a colleague of mine and I looked at this question, and we asked which methods are best for combining these. So how sensitive are these group maps to the effects of individual subjects? Okay, so what technique is the best technique to use? So we just briefly did this little experiment where we looked at 4 of these different combining data methods. We took 11 different subjects, and what we did is we made these what we called leave one out maps. So we recomputed these group maps 11 different times, each time leaving one of the participants out, okay? So we did this, we recomputed the maps, then we tried to get a quantitative measure of how much one individual subject affected our group results. And here's what we used for these to try to look at how much an individual affected the group map. So using these leave one out maps, and we compared these to the complete group map where we concluded all of the 11 subjects, okay? So again, we would do 10 subjects, leaving one out, and we'd compare this to using all 11 subjects. So what we computed was the number of enhancing voxels. So based on this leave one out map, we could look at how many additional voxels were active when we put this subject back into the 11 subject group map. We could look at how many voxels were diminished by putting this subject in compared to leaving them out. And we could look at the relative effect. So we took the number of enhancing and the number of diminishing voxels, and we divided this by the total number of active voxels in the complete group map. And then we looked at this percent overlap, okay? So again, this is a measure of how much leaving a person out of a map affected the overall group map. So it was a pretty simple-ish type of study, just to sort of get an idea. And here's an example of two different subjects. The difference maps. So this is how many voxels went away and how many came back by putting subject 6 in and putting subject 11 in. So these are the difference maps on the left, and on the right here are the overlap maps. So these are the red represents the number of voxels that were active in the complete group map, and the green is how much overlap was between a single subject and the complete group map, okay? So we did this when we wrote a paper on it, and we had these disparity tables, which this is the subject number 1 to 11 on the left. This is how many voxels became added to this complete group map by putting them in. This is how many voxels, the diminishing, went away when we put this person in. So the person can either add to the information so that it makes an active voxel, or they can drag away from that activation so that the voxel becomes non-active when we put them in. And then we looked at the relative effect and their percent overlap with the complete group map. So you can see here there's quite variation between the different subjects when we look at this. And this is for using the Stouffer method of group maps. And we also did this using the Fisher method and the random effects method. So just to see how much each technique really, how sensitive they were to the effect of a single subject. So you can see here, Fisher's technique really can change the number of voxels in the group map by adding or taking away a single subject. On the other hand, using the random effects model, we see that adding or deleting a subject did not affect this complete group map as much. So the conclusion from this study was that some of these tests that, especially Fisher's method, were very sensitive to the effects of individual subjects. And something like the random effects was the least sensitive to the adding and removing of subjects. And the averaging of T-maps, which we also looked at, and Stouffer's method, were somewhere in the middle. So based on this, we decided to make the conclusion that if you're going to do an exploratory study where you have a limited number of subjects, you might want to do something like Fisher's method, so you can really get an idea of what areas of the brain are active for different subjects. However, if you have a large group of subjects and are doing something that you want less sensitivity in your group maps. So it really doesn't matter that much if a person is added or left out. You might want to use something like the random effects. Okay, so that was the lecture on modeling the data. The next lecture that I have is just a general showing us going through each step in the data. So I think I'm going to start that today, unless everybody is really tired and wants to wait till tomorrow.